Good morning or good afternoon, everyone. I'm Julia Caro, Managing Editor at GenomeWeb, and I'll be your moderator today. The title of today's webinar is Antibiotic Gene Surveillance, What You Don't See Can Hurt You. The sponsor of our webinar is Streck. Our speaker today is Dr. Nancy Hansen, Director of the Center for Research in Anti-Infectives and Biotechnology and Professor in the Department of Medical Microbiology and Immunology at Creighton University. You may type in a question at any time during the webinar, and you can do this through the control panel, which normally appears on the right side of your screen. Just click on the Q&A box on the upper right side of the control panel, and when you click on Send To, please select All Panelists. We will ask our speaker your questions after her talk has concluded. So with that, let me turn it over to Dr. Hansen, who will now share her screen with you. Thank you very much.
Well, hello everyone. I'm glad you could join me today. I want to first thank GenomeWeb and Streck for giving me the opportunity to talk to all of you today. So the first thing I want to do is define antibiotic resistance. And you might think this might be an easy definition, but I want to make sure that you understand that it's the acquired ability of a pathogen to withstand an antibiotic that kills its sensitive counterparts. That means that a Pseudomonas susceptibility won't be the same as a Klebsiella, so we have to look at resistance from the point of the organism. So the first question I'd like to pose is how much and where are antibiotics being prescribed in the United States? As you can see from the U.S. map, the South is in bright red here, along with Nebraska, my state, and Iowa, of having the most usage in the U.S., whereas people on the East or West Coast and Alaska seem to use the least amount. And there's at least 30% of antibiotics prescribed in doctors' offices, emergency departments, and hospital clinics, which are unnecessary. And I just read a paper the other day which the clinics can be as high as 48%. And so that leaves 47 million unnecessary antibiotic prescriptions by U.S. doctors' offices and emergency departments, which we've got to change that somehow. 2 million antibiotic resistance illnesses and 23,000 deaths over each year. So in 2013, the CDC put out a threat level for resistant organisms, and they divided it into 3 levels. You have urgent, serious, and concerning. And carbapenem-resistant Enterobacteriaceae top the list here, or CREs. My lab is interested in CREs, multidrug-resistant Acinetobacter, multidrug-resistant Pseudomonas aeruginosa, and extended-spectrum beta-lactamase-producing Enterobacteriaceae, which are all in the serious level. The one thing I want to point out is that when this description of CREs came out, that stands for carbapenem-resistant Enterobacteriaceae, and now we use the term carbapenemase-producing Enterobacteriaceae because these are the organisms that produce the carbapenemase that molecular detection are looking for. These organisms actually can produce a carbapenemase, but they don't have to, and there could be chromosomal mechanisms that can contribute to that resistant phenotype. So if we take a step back and look at a world with no antibiotics, we'll certainly have to modify our medical care. And this includes, of course, treatment of bacterial infections. We don't have as many options as we used to. But think about all the patients that take chemotherapy every year. They all need antibiotics for that procedure to occur. Hip and knee replacements are the same, heart surgery, major GI surgery. And one thing I don't think people think about are transplants, kidney, liver, heart, bone marrow transplants. We know that in solid organs they look for viral infections, but they don't look for antibiotic-resistant organisms, and that needs to change. There are several publications out there now that talk about these organs being having resistant organisms in those organs, and so people need to be aware of that. The other thing I want to point out is the new movement of One Health. That has been in the last few years, and that emphasizes the health linkages among human, animal, and environment. And we used to be just, worried about human health, but animals, veterinarians, and ranchers use antibiotics, the environment, plants, etc. Wherever you look for an antibiotic-resistant organism, whether it be in humans, animals, or environment, you will find them. So somehow we have got to pull together and take this antibiotic resistance just out of humans or in addition to animal and environment. And here's how that occurs. We use antibiotics, both humans and animals, food-producing animals, but we give antibiotics to our beloved pets. And the food-producing animals, we can get antibiotic resistance in the food chain, but people excrete antibiotics into the sewer. Animals excrete it into the manure. Manure sometimes is used for fertilizer, if you will, or gets into the water and the soil, which then can then get into our produce. All of these are publications within the literature showing resistance in our fresh vegetables. It also gets into wildlife. And if any of you are beach bunnies and you like to lay on the beach, be careful of those seagulls, because in their fecal material they carry extended spectrum beta-lactamase producers. So wash your hands and wash your hair if the seagull happens to spat you down. So why are gram-negatives so difficult to determine, A, the resistance mechanism, and B, to design new antibiotics? We have new drugs for gram-positive organisms, but it's these gram-negatives that seem to be very problematic. Now that's because they have so many resistance mechanisms to choose from, and this slide will show that. So here we have a gram-negative organism. You can tell by the outside membrane and the inside membrane. And in between is called the periplasmic space, and that periplasmic space houses beta-lactamases. That's where they hang out. Now, a beta-lactamase can inactivate a beta-lactam drug by inactivating this beta-lactam ring. So when a beta-lactam enters the cell through an opening called a porin, if there's a beta-lactamase hovering around, it can bind to that beta-lactam molecule and render it inactive, so it cannot reach its target, the penicillin binding protein. Now, some of these enzymes, they don't need to be a lot of enzyme in the periplasmic space to inactivate a particular beta-lactam, but in some cases there are, and so organisms have designed a mechanism to produce a lot of beta-lactamase. And so that's mechanism number 2. With a beta-lactam, if the opening for entry becomes downregulated or it's not present anymore, that's another way the organism can become resistant. And so now we have at least 3 ways. Other ways are target modification. In the case of fluoroquinolones, the DNA gyrase and topoisomerase 4, which are involved in DNA replication, can be modified so the fluoroquinolone cannot bind anymore. And then you can have active efflux that will take the drug either from the cytoplasm and push it out of the cell, or from the periplasmic space and push it out of the cell. So some of these organisms have all of this going on, plus modification enzymes for aminoglycoside resistance. And then, of course, you have plasmid-encoded genes that come in and move around from cell to cell, and so there's another mechanism. And so when we talk about multi-drug or pan-drug resistance, some of these organisms produce all of these beta-lactamase resistance, or all of these antibiotic resistance mechanisms thus rendering any drug we would use ineffective. Another thing that people are starting to at least understand, but maybe not the mechanism by which it occurs, is synergism between all these resistance mechanisms. So let's say we have a beta-lactamase in the periplasmic space. All right, maybe it's maybe it only hydrolyzes some enzyme or some beta-lactams, but then you get a combination of a downregulation of this porin, so the drug can't come in anymore with the beta-lactamase. That gives a certain level of resistance. And then you have this activated. So each one of these contributes to the resistant phenotype, but at this time we don't know how much one contributes over the other. So what happens when you have beta-lactamase-mediated resistance? Well, we have a lot of beta-lactams to choose from. We have penicillins, cephalosporins, beta-lactam inhibitor combinations, monobactams, and carbapenems. But when you start getting these enzymes activated in a cell, you can lose the penicillins. So then the physicians only have the rest of these to choose from. Then if you have an ESBL, you lose the cephalosporins. You lose the beta-lactam inhibitor combinations with AmpC beta-lactamases. You lose the carbapenems when you have a carbapenemase. And the two new drugs that are out on the market that came out, I believe, in 2015 now have some resistance occurring. So it's possible that a physician can lose the largest drug class they have to work with these resistance mechanisms. So the beta-lactamases are the most common mechanism of beta-lactam resistance in gram-negative bacteria, and actually there's over 2,000 different types. They can be divided by substrate profile, which was first published by Bush, Jacobi, and Medeiros in 1995, and they group them into 4 different groups. Then you can divide them molecularly, and they happen to fall out in the same groups. These include the AmpC beta-lactamases, the Class A, which houses ESBLs as well as Class A carbapenemases. You have the metallo-beta-lactamases, and then you have the Class D enzymes, which include penicillinases, which when I started in this field 23 years ago, that's all we worried about, and now they have evolved into ESBLs and carbapenemases. So the evolution of antibiotics and resistance kind of go hand in hand. With not much after the introduction of penicillin, you saw penicillin-resistant Staphylococcus aureus occur, and then as we progress, we saw an explosion of all of these different beta-lactamases, and that occurred after the introduction of cephalosporins and then carbapenems. So beta-lactamase type differs, and that difference allows different hydrolytic properties for those enzymes. So for example, broad-spectrum beta-lactamases will hydrolyze the penicillins, where extended-spectrum beta-lactamases will hydrolyze the cephalosporins, but they can also hydrolyze a lot of the penicillins. AmpC beta-lactamases can interfere with the beta-lactam inhibitor combinations, such as amoxicillin clavulanic acid, otherwise known as augmentin, ampicillin sulbactam, piperacillin tazobactam, and ticarcillin clavulanate. But these enzymes can also interfere with the cephalosporins and the penicillins, and then you have the carbapenems. So the carbapenemases sometimes can inhibit every beta-lactam drug class we have. The new ones, Zerbaxa, otherwise ceftolozane tazobactam, can inhibit TEM, SHVs, CTXMs, which are the ESBLs, some AmpCs, but not all of them, and some Class Ds, but not all of them. ABCAS can inhibit KPC carbapenemases. So let's go over some of the main families. So the main families containing ESBLs include TEM, SHV, CTXMs, and OXs. Now the CTXMs, no matter which CTXM you have, they're all ESBLs. So there's no trying to figure out which ones are broad spectrum versus extended spectrum. But with the TEM and the SHVs, some of these the majority are probably ESBLs, but then you have broad-spectrum SHVs and TIMs. And so if you're using a universal primer set, for example, you won't be able to tell if you have a TIM or SHV ESBL or just a broad spectrum. In fact, Klebsiella pneumoniae carries SHV1 on its chromosome, so 99.9% of all the Klebsiellas will be positive for an SHV gene. For the OXs, the only OXA ESBLs you will find, well, I should say only, bugs have a tendency to surprise us, but the majority of them are found in Pseudomonas. If you're looking at phenotype or susceptibility, the substrate preferences for these ESBLs do vary. So you can have a ceftazidimase, a cefotaximase, or you can have enzymes with no preference. And so by picking one substrate, you won't be able to detect all the ESBLs that are out there. And in addition, TIM and SHV used to be the most prevalent ESBLs in the world, but now it's the CTXM class, and specifically CTXM15 and CTXM14. And here's a website that if you're interested in looking at all the different 2,000 beta-lactamases, you can access this site. So who carries these ESBLs around? Well, ESBL-producing organisms that are covered by the CLSI, or the Clinical Laboratory Standards Institute, are E. coli, Klebsiella, and Proteus mirabilis. They have criteria for finding ESBLs in these organisms. The problem is that all of these organisms, such as Citrobacter, Enterobacter, Morganella morgani, and etc., they too can carry ESBLs, which are not covered by the CLSI, so you could have those for example, in your hospital, but because you're not looking for them, they actually act as undetected reservoirs of ESBLs. And until a plasmid carrying the gene for an ESBL moves from Citrobacter to an E. coli, you won't find it. One approach by the CLSI for a confirmatory ESBL test is you can use BROF or disk diffusion. I have an example of disk diffusion here where you plate the organism of interest and then you use ceftazidime plus and minus clavulanic acid, or cefotaxime plus or minus clavulanic acid. And if the zone of inhibition increases by 5 millimeters, it's pretty certain you have an ESBL. And obviously you can see that the bug grows right up to the disc here, but in the presence of clavulanic acid, you get this nice big zone size. So now let's move on to AmpC beta-lactamases. These are called Cephalosporinases. They're resistant to beta-lactamase inhibitors, and some of these enzymes are inducible. So that first slide I showed you where all of those beta-lactamases went into the periplasmic space, that can happen by induction. So there's lots of different AmpC players. We have AmpC beta-lactamases that are encoded on the chromosome, such as Pseudomonas and Enterobacter and other species. These are inducible, whereas E. coli also has a chromosomal AmpC, but that gene is not inducible. So they cause resistance by a different mechanism as far as overexpressing that gene. Now, in the late 1980s, some of the genes that were on the chromosome moved on to plasmids, and there are 6 families of plasmid-encoded AmpCs, CMY2 being the most prevalent globally. Each one of these families has derived itself from a chromosomal organism. So CMY2 came from Citrobacter fundii, DHA is prevalent in Asia and in some U.S. states. It's inducible. Most plasmid-encoded AmpCs are not inducible, but the ones that are more concerning to me, and I'll explain that when we get to surveillance. ACT1 is from Enterobacter. These two are from different species of Enterobacter. ACT1 is inducible, MIR1 is not. FOX and MOX come from 2 different species of Aeromonas, and then ACC comes from Hafnia alvei. The problem with the naming of these plasmid-encoded AmpCs is that people have named them CMY even though they're from two different genera, and so it gets very complicated out there. The naming needs to be changed. So what organisms produce chromosomally inducible chromosomal AmpC cephalosporinases? Well, you have Enterobacter, Serratia, Citrobacter, Ferndia, Proventia, Morganella, Hafnia, Pseudomonas, Aeromonas, and Yersinia intercolitica. Now, these types of organisms have specific induction pathways that can give them the ability to increase the amount of AmpC enzyme in the presence of certain drugs. How does that happen, and why is that clinically relevant? Well, some of these bugs can mutate and cause a huge amount of AmpC beta-lactamase being produced all the time, not just during an induction phase. And we call those Derepressed mutants. So if you take an infection in a patient and you have a bacterial population prior to treatment, it might look like this, where some of the organisms in that population, even though they're all, say, Enterobacter cloacae, some of them are going to be mutated as a Derepressed mutant, so expressing a lot of that beta-lactamase, that AmpC beta-lactamase. This happens in about 1 in a million cells. If a physician were to treat with a third-generation cephalosporin, that drug will kill the wild-type cells, but not the Derepressed cells. So actually what's happening during therapy is the selection of these Derepressed mutants. And 25% of the time, the patient will fail therapy because the Derepressed mutant takes over the infection. So that's clinically relevant. Now with plasmid-encoded AmpC beta-lactamases, you have a different scenario sometimes of how this overproduction of the AmpC beta-lactamase occurs. These genes are found on plasmids, so depending on how many copies of that plasmid you have in a cell will depend on the level of beta-lactamase found in the periplasmic space. If for some reason you have more copies than in another cell, you can get more beta-lactamase in the periplasmic space. If that enzyme is inducible, like DHA, you can even get more in the presence of certain drugs like cefotaxime and imipenem. So the issues with these plasmid-encoded AmpC enzymes is that in the clinical lab, you'll get false susceptibility. They'll look susceptible on a plate, but the AmpC gene is there and has the propensity to cause resistance in the patient as well as carbapenem resistance. So you can get multi-drug resistance. Detection is a problem in the presence of an organism that has both an ESBL and an AmpC. You typically don't find the AmpC, so the AmpCs out there for prevalence are undercalled. About 3% of E. coli carry plasmid-encoded AmpCs, and if you think about all the E. coli infections, that's a lot of AmpC beta-lactamases. Infection control is another issue, and development of carbapenem resistance with that downregulation of that porin. You can test for this by using Cefepime, which it works against AmpC-producing organisms, and then Clavulanate to inhibit the ESBL. So this organism has a CTXM ESBL and a plasmid-encoded AmpC. The Cefepime you see is resistant in the presence of the ESBL, but if you use Clavulanate, you can see a zone indicating the presence of an AmpC. So AmpC confirmatory tests you can demonstrate that with sulfoxetine hydrolysis, or you can inhibit the AmpC gene, or you can detect it using genetic tests, multiplex PCR. Phenotypic tests are reliable if the organism produces a decent amount of AmpC beta-lactamase, and a lot of these organisms produce different levels, so there's an issue there. AmpC DIS tests for phenotypic tests, and AmpC inhibitors like boronic acid and cloxacillin. If you want to look for plasmid-encoded AmpCs, the best way to do that is with the multiplex PCR. So this is an example of an AmpC disc test. You have a culture of very sensitive organism on the plate. You have a sulfoxetine disc, which can induce the beta-lactamase, or if it's a plasmid-encoded one, it is typically resistant. Then you take your organism of interest and you smear it on this disc that has EDTA, allowing the enzyme to leak out and protect the sensitive bug here, the sensitive E. coli, decreasing the zone on that side, indicating the presence of a AmpC beta-lactamase. But with this test, since it's phenotypic, you cannot differentiate between chromosomal and AmpC beta-lactamases. To detect an inducible AmpC, you can use the disc induction test, both for chromosomal and plasmid-encoded. The plasmid-encoded, there's only a few of those, as I mentioned, DHA, ACT1, CFE, and CMY13. For that test, you plate the organism, the test organism on the plate. You put a sulfoxetine disc here, which is involved in induction. So it's going to induce that AmpC gene if this organism carries it. You put 2 discs of antibiotic. This happens to be piperacillin and tazobactam, and as TRINM, that you show some sensitivity. But if you get induction, you get a flattening of the zone because a sulfoxetine has allowed these organisms to induce its AmpC beta-lactamase. So now let's move on to the carbapentamases. There are several kinds of carbapentamases. Plasmid-encoded class A's, the most recognized one is KPCs, or Klebsiella pneumonia carbapentamase. There's also an increase in GES genes. They're found all over the world. They're not found very often, but you have to ask yourself, are they rare because they're not there, or are they rare because we can't find them? Chromosomal carbapentamases we also see. ME and NMC are found in Enterobacter. ME2 actually has moved on to a plasmid, just like the plasmid-encoded AmpCs, and they are inducible like inducible AmpCs. Serratia marcescens is beginning to be more of a concern. It has the chromosomal beta-lactamase, and then attached to it is the gene that helps induce the amount of enzyme that you see in the cell. And then these are you don't see these organisms too often, but they do have an inducible chromosomal carbapentamase. We have class B enzymes known as metallobeta-lactamases, the VIM, the IMPs, and the NDMs. And then class D are the oxocarbapentamases. Oxa48 is found in Enterobacter acae, both E. coli and Klebsiella. These oxas, 23, 24, 40, 58, and 143 families are typically found in Acinetobacter. And then Acinetobacter oxa51 is found on the chromosome of Acinetobacter, but in some instances it has moved on to plasmids. So this gives you a world view of all the carbapentamases out there and their impact in different countries. So if you look at the different markings, you see that a solid mark indicates an endemic or nationwide distribution, and then these circles here represent significant outbreaks or a regional spread, and then sporadic occurrence. So if you look at, for example, Japan has a real problem, an endemic with IMP beta-lactamases, whereas India has NDM problems, and let's see, Puerto Rico has KPC problems. We have KPC problems in the U.S., and every state now in the U.S. has identified a KPC-producing organism. If you take a closer look at Europe, Greece has endemic problems for IMPs and KPCs and VIMs. And so this just gives you an idea of how widespread these carbapentamases and all of these beta-lactamases are in the world. The newest threat we have is not a beta-lactamase, but colistin resistance. And typically it used to be found on the chromosome, and so you didn't see very much of that. And then in the late 2016, the Chinese were doing a study and found what they named a plasmid-encoded MCR1 gene causing colistin resistance. What this gene product does is it modifies lipid A, causing resistance. They first found it in pigs and chickens because the vets were using it, and then they found it in humans, and now we can find it all over the world. Why? Because we know what to look for. The one thing about molecular detection is you have to know what you're looking for. If you don't, you can't find it. So this just gives you an idea of the complexity of colistin resistance. You have lots of relay pathways here. One pathway relays these genes, which relay these genes, which relay these genes, and these genes are involved in modifications of the lipid outside, the LPS. This is the gene that moved on to the plasmid that is now known as MCR. And this is an example of a plasmid. It's 64,000 base pairs. Here's the MCR1 gene. But there are different plasmids carrying MCR1 all over the world, and a lot of them are associated with beta-lactamase genes, NDM, carbapentase. I haven't seen KPCs, CTXMs, and also plasmid-encoded AmpCs are found in addition to MCR, thus giving you multi-drug resistance with one plasmid. So, if I haven't convinced you yet, I hope I have, that surveillance is really the key to helping us slow down the emergence of resistance. You're never going to stop it totally, but you can slow it down, and it has been shown to work. So the current detection methods are still mostly phenotypic, DISC, microbroth, automated susceptibility. Molecular tests are out there for these beta-lactamases. Some are FDA cleared, some are not. The difficulty with phenotypic testing for ESBLs, AmpCs, and carbapentamases is that the susceptibility patterns can be highly variable. With all of those different mechanisms being produced all at different levels, you can see how that variability might play havoc with phenotypic testing. The other thing is that there's multiple mechanisms of beta-lactam resistance and multiple beta-lactamases within one organism. We published a paper where there was at least 8 different beta-lactamases that were actually produced. We detected them by IEF, isoelectric focusing, and the organism produced 8 different ones, and we saw a couple that we couldn't identify. So they're out there. So what are our options when it comes to molecular testing? Well, we can go after the gene. We can go after the expression of the gene by looking at RNA. We can go after the gene product, which is protein in most cases. Right now, the tests go after DNA. It's low-hanging fruit. DNA is more stable than RNA, and it's easier to design assays for. So at the present time, what we should look for are the CTXMs, because they're both in hospital and community-acquired organisms. Imported AmpCs, those 6 families I mentioned, CMY2, DHA, Interbacter, Fox-Mox, and ACC, and the carbapentamases, KPCs, the metallos, VIM, IMP, and NDM, and OXAs, OXA48, and their family members. And now the newer threat, MCR gene should be thrown in the mix. So why do I think we need to target these beta-lactamases? Well, actually there are 5 genetically distinct classes of CTXMs, but the most prevalent are CTXM14, which are in group 9, and CTXM15, which are in group 1. These are the most prevalent worldwide, and CTXM15, although the data is limited, seems to be associated with complicated UTIs or urinary tract infections and urosepsis. Then we have the AmpCs. There are no recommendations for testing by the CLSI for AmpC beta-lactamases. That's why they're underrepresented. That's why they're missed in phenotypic tests. When they are detected, they're mistaken as an ESBL, and when they go undetected, there was a nice paper published by Pai et al. in Antimicrobial Agents and Chemotherapy in 2004, associating the lack of detecting a plasmid-encoded AmpC with therapeutic failure. And then the carbapentamases. Metallo-beta-lactamases are resistant to all antibiotics except aztreonam, and it's rare now that you find a carbapentamase alone in a cell, in a bacterial cell, so you can't even rely on this anymore. And then there are always exceptions to the rules out there, and in Enterobacteriaceae, sometimes they're difficult to detect phenotypically. Class A carbapentamases, the KPCs, are found in Enterobacteriaceae, and since they've lowered the breakpoints, you don't miss as many in the clinical laboratory, but you can still miss some. And then the Class D carbapentamases, OXA48, they're found in Enterobacteriaceae, Pseudomonas, and Acinetobacter. In fact, OXA48 was first found in a Klebsiella pneumoniae in Turkey, and these are not always detected by susceptibility assays. So the only experience I've had is with the Streck ARM-D Kit, and so I will talk about that for a moment, but there are other tests out there. In this particular test for, well, not for AmpCs, there's not really, but the Streck ARM-D Kit for AmpC has 2 PCR mixes. Each target is with a different fluorescent dye, and as you can see in this example, for the target of the Fox beta-lactamase, these red lines represent different clinical isolates. You can see that when they're negative, there's no getting them mixed up with a positive isolate. And then these yellow lines here represent the positive controls for that particular gene that's included in the kit. So they can detect CMY2, DHA, the blue lines here, and then every kit has an internal control just to make sure that the DNA that you isolate and that you are testing is capable of being amplified in a PCR reaction. The other kit, ARMED kit, detects 9 different beta-lactamases in 3 PCR mixes, the CTX-Ms, both 15 and 14, being the most prevalent ESBL in the world, the Class A carbapentamase KPC, which is endemic in many countries and all over the world as well, OXA48, for example, and then again an internal control. And each one shows the positive control here. So in summary, rapid surveillance is key in my opinion. It's key for infection control, for antibiotic stewardship, and for optimization of new drug use. And that's key with the new drugs that are in development and coming onto the market for gram-negatives. We've got to be able to find those resistance organisms so that we can optimize the drug use. But each hospital is going to be unique as to what they need to do and what type of detection they're going to use. Routine surveillance is best, in my opinion. Different wards, though, might require different surveillance, different frequencies, weekly, monthly, hopefully before a large outbreak occurs. And then no single test will suffice. Both phenotypic and genotypic tests are going to be required. Again, the genotype is only if we know what it is, but a phenotype can help us determine if something is emerging. And last but not least, if I've intrigued you with any of the information, I will make a plug for Creighton University. I teach a course in the summer in May. It's 2.5 days, and if you're interested in learning more about any of this, you can check out our mini course on antimicrobial agents and chemotherapy at Creighton University. And with that, I'll turn it over to Julie. Thank you for listening.
Thank you, Nancy, for this really nice presentation. As a quick reminder, if you have a question, please type it into the Q&A box on the control panel. Just before we enter the Q&A, we'd like to ask our attendees to take a brief moment after the webinar has ended to take our exit survey and give us your feedback. So with that, let's start with the Q&A. And joining us for this is Stephen Kelly, who is Product Manager at Streck.
Hello?
Hi. So here's a question probably for Nancy. Can the beta-lactamase interfere with the identification of gram-negative pathogens?
Not that I'm aware of, because those IDs are usually not dependent on whether or not that organism has a beta-lactamase. And not all gram-negative bacteria will carry a beta-lactamase. So I would say no.
Okay, thanks. Another one probably also for Nancy. Does the resistant gene expression and the effect on antibiotic prescribing apply to all body fluids tested, for example, blood, sputum, stool, or others?
Well, that's a good question. Probably. Everything that we do, you know, is in the laboratory. So, and we can't test it at the moment. I'm not aware of anything that we can use to test it directly out of bodily fluid. We can detect it, and some tests can detect it, right? But you'd have to measure the level of protein or the level of RNA maybe, and those tests just aren't out there.
Okay, thanks, Nancy. What would you say are the biggest limitations of current phenotypic testing, and are there any advantages over molecular testing?
Well, the limitations of phenotypic testing is become clear when the organisms produce multiple multiple resistance mechanisms, and so if a certain resistance mechanism is associated with therapeutic failure, like the KPC carbapenemase, if you just identify the fact that it's a carbapenem-resistant organism, you don't know what's causing that, where a molecular test will help differentiate the cause or the contribution of a particular resistance mechanism to that phenotype. So I would say that both tests have limitations, and that's why I think really strongly that both tests need to be run. Now, in trying to get the quickest answer, if you were to do a molecular test for a KPC producer, for example, first, and then follow it up with a phenotype, you would know if it had a KPC gene that is probably not the best to try and treat with and there's lots of controversy out there, and I'm not advocating one thing or the other, but maybe you would rethink treating with a single carbapenem, so to speak. Some doctors would never treat with two carbapenems, but some would treat with a carbapenem and an aminoglycoside, so there's lots of things to think about, but at least you know what you're dealing with.
Great, thank you. A related question that's a little more specific, and that is the Kirby-Bauer test still a useful test for routine surveillance in hospitals?
Well, I know that a lot of hospitals use them for urines, for E. coli, and it all depends on who's reading the test, really. If you've got an experienced, and first of all, I want to make sure that the audience knows that I do not run a clinical microbiology lab, I run a research laboratory. But from what I'm told, if you use Kirby-Bauer, sometimes if you have an experienced person reading those plates, the zone size might change even though it doesn't relate to a resistant phenotype or a specific criteria, you can see maybe something happening in your patient population with a Kirby-Bauer. But you got to have some experience with that. But I like Kirby-Bauers. And I know that we use them here at Creighton, so for urines in particular. So that's all I got on that one.
Good, thank you, Nancy. What would you say are the best clinical databases where you can find gene sequences of common resistance-related enzymes?
Well, NCBI has taken that over, and on that first ESBL slide, I have the website there, and that's what is recommended now.
Okay, thanks. A specific question from one of our participants about their project. This participant has an interest in detecting antibiotic resistance genes in clinical wastewater. And the issue is that the participant doesn't know which of the gene he or she should focus on and which method should be employed. Do you have any advice on that?
Well, let's see. Since CTXM ESBLs are the most prevalent worldwide, I would certainly look for those. They will find TET-resistant genes and chloramphenicol resistance genes because a lot of these organisms just now carry them around. So if you want to look for something that's pertinent to treatment, you know, the major drugs that are used for treatment these days, I would look for the methyltransferase for 16S ribosomal that gives rise to aminoglycoside resistance. I would look for beta-lactamase genes like CTXM, all 5 families, but depends on what region of the world you're at. All beta-lactamases seem, well, not all, but there's some beta-lactamases that are found that are more prevalent in some countries than others. Like CTXM2 is found in Argentina, where we don't see it a lot in the U.S. And then for the carbapentamases, depends on, again, what part of the world you're at. But if you were in the U.S., I would look for KPCs, or if you're in Puerto Rico or Israel, they have a high incidence, or they used to, of KPCs. If you're in India, I would look for NDMs. It all depends on where you're at. And that map that I put up there might help you, at least for the carbapentamases. So did I help this person enough?
I think so. Thank you. Yeah. Other question you may know about, what about multi-TOF detection? Do you know, can you comment on that?
I don't know much about that. I know it's been used for species identification, and they've also used it to determine if there is a carbapenemase in there, but they have to use imipenem, and then they look at the when the enzyme cleaves the beta-lactam, there's 2 different peaks or 3 or something that show up, so that somebody used to reading that type of data can discern whether or not the drug has been cleaved by a beta-lactamase, a carbapenemase. But that's all I've got on that one because I have never worked with that, so I hate to comment any longer or any further.
Thanks. Question about point-of-care testing. Do you think that should or could replace the current standard, and how should it be used? Point
of care, I don't think it's ever going to replace what's done now in the hospital. I think what point of care could be useful for is doctor's offices, just like a general strep test or an influenza test, because if we could figure out, or if the physicians could have a clear understanding of well, there's nothing here that we need to give an antibiotic for, that would be a huge step. And also in the emergency room. Those are the two places where the highest amount of antibiotics are given that are not necessary. And so I think point of care would be great in those two locations for sure.
Okay, thanks. Are beta-lactam drugs ever used in combination as a multi-drug therapy?
Oh yeah, they're used with aminoglycosides all the time. So, and then it just depends on the organism, the resistance mechanism, and the patient's health, really, because if they've got kidney problems or stuff, then it's harder to use an aminoglycoside, but sometimes the physician has no choice. So you have to understand your patient, the bug that you're, or the bacteria you're trying to treat, and in my opinion, the resistance mechanisms that that organism carries, so that you don't waste the patient's time trying to find the right combination. So if you know those three things, then the guess that you use is the best guess you have, right?
Okay, thanks. Here's a request for collaboration. So somebody is asking whether you have any genotypic surveillance system implemented in your lab and whether it's possible that this participant can collaborate you for developing such a system in their own country.
The answer is yes and yes. So have them email me and we'll talk about it. Make sure that when you do email me, I talk to a lot of people, and just tell me where we met, so you can say Genome Web, and then we'll talk about how I could help you do that, because I think surveillance in every country is necessary. You could also talk to Streck, who is willing to, they find that, from my point of view, important as well. So you might have a connection there as well.
Okay, a specific question about one of the assays. Does the CARBA-R assay cover sufficient types of carbapenomases that are currently found in the US in clinical samples?
I don't know if I can comment on that. I've never used the CARBA-R, and I have hearsay, but I don't know for sure, so I just assume not comment on that.
Fine. That's fine. Thank you. Question about whole genome sequencing. Is bacterial whole genome sequencing replacing screening of mutations in surveillance programs?
Well, I think whole genome sequencing has a place, but it's slow, and in the clinical lab, we really don't need all that information. For surveillance, whole genome sequencing gives you interesting data, but finding mutations in different genes will only tell us there's mutations in different genes. Now, if there are mutations in genes that we know are involved in resistance, for example, in Pseudomonas, the porin OPRD, when it is downregulated and you have an insert or something that disrupts the reading frame, then you can pretty much figure that it's going to be imipenem resistant. But in genes that we don't know what the function is with regards to resistance, those things just still need a lot of basic science to get that information so that it means something, in my opinion.
Okay, thank you. How would you describe the level of CARBA or MDR awareness in the US? Is it a real concern for hospital administrations, for example?
Well, I don't talk to hospital administrators probably at all. I wish I did. I think everybody is aware of it as far as in the lab. But whether administrators find a need to, you know, the almighty dollar is still a concern, right? And it depends on, like I said, every hospital is going to be different. If they're looking at phenotypic surveillance and they don't see a lot of resistance, then they're probably not too worried about it. And there's been labs in the Midwest anyway, or the Plains where I'm located, that when they have a concern and they can't figure it out, these smaller hospitals and laboratories, they'll send me the bug and I'll figure it out for them, but they don't have very many of those. So
doesn't make any sense.
I suppose it doesn't make a lot of sense to put a lot of money in it if where you're at doesn't seem to have a lot of resistance, which is probably a good thing. It is a good thing, but becoming more rare. You just have to watch what your organisms are doing by your antihistograms.
Right,
or
thank
antibiograms, I'm sorry.
Okay. Is surveillance data added to the resistance map database? Do you know?
That I don't know.
Okay. And we have one more question. Could phenotypic tests become rapid enough to impact clinical outcomes in treatment decisions that are time sensitive? And what technologies are coming that address this need, for example, in sepsis?
Can answer that last one. And the thing with the phenotypic test is you still have to grow the organism. You have to isolate it, and you have to grow it. So depending on how fast that can happen will determine on how fast you can get an answer. But in my understanding, you still have to, you know, if you have a mixed culture and there's other organisms in that population, you've got to find the one that's causing the problem. And there are ways to do that, but I don't know how quickly that can be done rather than what it's being done now. But that's not my expertise, so I could be mistaken. Good question, though.
Okay. Do you think it's important to accompany the launch of a new antibiotic with a diagnostic test to identify resistance mechanisms associated with that drug? Can you just comment on that?
Absolutely. I think that the reason we're in a fix like we are is because no one was looking for ESBLs, even though some people were, but nobody was listening. And so now we have this problem of cephalosporin resistance everywhere. And with these new drugs coming out, a lot of them are similar in nature. And so the organisms are going to develop resistance. And if we're going to use those optimally for both the patient, the amount of money a drug company puts into developing that drug, we need a diagnostic to go along with it. And we need to start looking for those resistance mechanisms as soon as possible. I mean, when AVCAS came out, everybody was excited. KPC wasn't going to, I mean, it was great for KPC producers, and yet the bugs have found a way to be resistant. Not a lot out there, thank goodness, but it happens. So we need to be able to find it. I feel pretty strong about that, could you tell?
Yeah. Here's one more question that just came in, and that is, as a clinician, do I need to be concerned about the genotypic surveillance data in the country, in the state, with reference to my own clinical practice?
Yes, the answer is yes. And I know that's a bummer to hear because you physicians have lots of things to keep track of. But if your state health department is doing their job, it should be easy data to find. And yet every patient population, even within a city, is going to have differences. So, but yes, I can't stress that enough. And I know that the days of SI and R are over. Sorry.
Okay, so This is actually all the time we have today for questions. Thank you very, very much.
Thank you.
Dr. Hansen. So we'd like to thank you and also our sponsor, Streck. And if we didn't have time to get to your question, we will try to ask Dr. Hansen to address it afterwards. As a quick reminder, please look out for the pop-up survey after you log out and provide us with your feedback. And finally, if you missed part of this webinar or you would like to listen to it again, we will email you a link to an archived version. So with that, thank you very much again for joining us for this genome webinar.